Welcome to Ash Jottings. In this video, we'll talk about DNA replication. It will be just an overview of DNA replication and what are the basic rules and requirements for DNA replication. So let's get started. So overview, let me just write it down. Overview of DNA replication. So it was, as we all know, it was Watson and Crick who suggested a very simple mechanism of DNA replication on the basis of its double helical structure. So during replication, what happens? As we all know, during replication, the hydrogen bonds which are present between the nitrogenous bases are going to break when these bonds are going to break. The double helical structure, the strand is going to get separated, right? So that now the polynucleotide strands of DNA separate, or you can say uncoil. Now, these separated strands are complementary to each other. Now, each of these separated strands will act as template for the synthesis or polymerization of other strand, right? Now, as the end, what will happen? There are two daughter DNA, two identical, let me add, identical DNA molecules are formed. So many things are going to happen in between. This is just an overview, right? Now, because of its specificity of base pairing, each nucleotide of the separated strand will attract its complementary nucleotides from the cell cytoplasm. Now, once the hydrogen bond, after it's formed, okay, hydrogen bonds are attached to nucleotide, what will happen? The next step will be the sugar, so which is going to join to their phosphate, completing the formation of, which will complete the formation of new polynucleotide chain. So we know that each strand, the separated strands, if this is a double, okay, let me just, double helical structure, okay, very bad diagram. Yes, it is going to get separated. Here you'll have, let me just, oh my God. Okay, let me just wipe this, and here. You can see the daughter strand will be like, and here also it will be like this. Let me just join this two. So this is how it is going to look, right? The two strands are getting separated because the hydrogen bonds are getting separated here, and this strand will act as this parental strand, is going to act as the template for the synthesis of this daughter strand. And similarly here, this strand is going to act as the template for the synthesis of this daughter strand. So this kind of replication is usually known as semi-conservative. Why it is called semi-conservative? As you can see, the daughter strand, it is a hybrid conserving one parental strand and other newly synthesized strand. Yes, so this method of replication, which usually occurs in double-stranded helical structure, is semi-conservative. Okay, now let's talk about the basic rules of DNA replication, which I'm sure all of you must be familiar with. Basic rule of DNA replication. And so you know that specificity plays an important role here, because the specificity of base pairing during replication is very important. That is, adenine is going to base pair with thymine and guanine base pairing with the cytosine. So the DNA polymerase, which is a very important enzyme in case of replication, is going to add nucleotide monomer one by one at 3' end of growing strand. Yes, and also you can say the nitrogenous bases of each daughter strand being formed is complementary to the template. We already spoke about it, right? So the nitrogenous nitrogenous bases of each of daughter strand will be complementary or should be complementary to template strand. And also we all know the 3' end, which has OH group, it will go and bind to another nucleotide, right? And the 5' end, which has phosphate group, it is going to bind or going to connect with the newly synthesized new polynucleotide chain synthesized in 5' to 3' direction. Yes, we'll talk about it. There's so many things going around. It is just a brief talk about DNA replication. Now, talking about the requirements for DNA replication, we'll talk one by one. Let me just write it down here. Requirement of DNA replication. If I have to say, the first one will be the precursor nucleotide molecules. What are precursor nucleotide molecules? There it is none other than the deoxyribo. Okay, let me just use a brush page. Yes, let me just write it down. Now, talking about the requirement for DNA replication, first one is the precursor nucleotide molecule. So what are they? They are deoxyribonucleotide triphosphate, nothing but DNTPs. We learned about all those things. So it is DNTPs, that means you need DATP, DGTP, DCTP, and DTTPs. Yes, you require this. And see, the three phosphate groups of this nucleoside triphosphate usually are designated as alpha, beta, and gamma. And it is to the alpha phosphate, that one is directly, it is going to attach to the deoxyribose. Okay. So the phosphates are of three types: alpha, beta, gamma, and alpha is the one which is going to get attached to the, or bind, it is going to get attached to this deoxyribose of this DNA structure. So talking about the second one, very obvious, you need a template DNA strand. A single strand of DNA is needed, which determines the sequence, right? It is going to determine the, let me write it down here, determine the sequence of nucleotide in the new strand, isn't it? New strand during polymerization. So it is very important. And then you require RNA primer. So we require RNA primer for the initiation of polymerization. Initiation of polymerization of new polynucleotide strands, right? Then you require, I'll talk about this in a while, and you need certain enzymes like DNA polymerase, and we talk, that is primases. Then you need ligase, helicase, then you need certain proteins for the proper functioning of DNA replication. There are certain proteins which are involved, and basically they will help in, let me just write it down, it will help in identifying origin of replication Then you need proteins for the recognizing the initiation point. Third, you need it for unzipping unzipping of nucleotide strands. Then you require protein for stabilizing this unzipped strands. Then you require recoiling. Yes, we'll talk about all these things. Recoiling of daughter strand. So these are the requirement for the DNA replication to occur. Now, talking about this RNA primer, as I told you, I'll come back to this. So usually when we talk DNA polymerase 3, this enzyme, it actually helps in the elongation of new polynucleotide, but it cannot initiate the polymerization. It cannot, it cannot initiate the polymerization. That is, cannot add the first nucleotide to start the synthesis of the daughter DNA strand. And that is, it can add nucleotide only when a chain of nucleotide is already passed. Okay, so for the initiation to occur, this DNA polymerase, especially in the lagging strand, it requires, that is known RNA enzyme known as RNA polymerase, or you can say it as primases. So what are RNA polymerase or primases? They are short RNA oligonucleotide segment formed by a sequence of 10 RNA nucleotide, which are basically complementary to parental template strand. So this DNA polymerase is going to recognize this RNA primer and adds DNA nucleotide to it, to its 3 end, and to construct a new DNA strand. So that is how, then once it has done, this RNA primer gets cleaved off and is replaced by DNA nucleotide. These are the things which you should remember before we actually talk about DNA replication. So these are the requirements. So let's come back. So DNA replication is a process in which new copy of DNA is produced from parent DNA. When two strands of DNA are separated, each strand is going to act as a template for the formation of new strand, right? And this process is called as replication. So let me just explain it once again with this diagram. If you can see, this is a double-stranded helix now with 5' and 3' end. So Initially, the first step is that you'll have helicase, which will help in the unwinding of this parental double helix. So once it is unwinded, the tension creator here will be actually controlled by certain proteins which are called as single-stranded binding protein, which will stabilize the unwound parental DNA. So this whole thing, you can see this is called as replication fork. Now, you will have one strand here. This is going to synthesize one new daughter strand, and this is going to synthesize another new daughter strand. So the leading strand is synthesized continuously from 5 prime to 3 prime direction by the DNA polymerase, and in this case only a single RNA primer is required. As it is a leading strand, there is no discontinuation. But when you see this strand, it is called the lagging strand, is synthesized discontinuously, and the primase synthesize a short RNA primer which is extended. So now you can see this is the RNA primer. It is going to initiate the synthesis in this lagging strand, and once it starts adding certain nucleotide, the DNA polymerase is going to come to the action, and it is going to it is going to add more nucleotide, and it is going to help in the elongation of the strand. So once all these strands have been synthesized, now who will come into action? That is the DNA ligase is going to come, and it is going to join the lagging strand. These sections will be joined here, right? So overall, the direction of replication is always 5 prime to 3 prime. Do not forget it. After the RNA primer is replaced by the DNA by another DNA polymerase, not shown, the DNA ligase join the Okazaki fragment to the growing strand. This is the summary or the overview of replication. Okay, so this type of replication is known as semi-conservative replication. Talking about this, let's talk what are the different types of replications. Okay, there are basically three modes of DNA replication. So one is the semi-conservative, which we just saw. And the next one is the conservative replication. And then you have the dispersive replication. So semi-conservative replication, here as we have seen, okay, I'm just going to give a brief again. So here the replicated DNA in the semi-conservative, just write it down. So here the replicated DNA molecule, it consists of one old plus one new strand, right? That is, old is the parental strand and new is the daughter strand. So the DNA unzips its two polynucleotides separately, and new complementary strand is synthesized on each two parental template strand. We just saw it. Now, this method faithfully copies the genetic information of each cell. So as and when you are going to replicate, how it is going to form, it will be like So basically you have a parental strand and this is the newly synthesized strand. So this method or this type of replication is known as semi-conservative method of replication. Next, talking about the conservative. So here the two newly synthesized polynucleotide strands are brought together. Strands are brought together and this is going to join to form a new DNA molecule. So what will have here, the parental strand reassociate and they form the parental DNA molecule. And the original DNA helix retains its, okay, let me just put it on. Okay. Yes. You have strand which is completely parental. So this is what I meant by reassociate. Okay, the parental strands are going to reassociate after the replication, and the daughter strands, which I'll just write like this, will be together. That is, there will be no mixing up. Now, the two parental strands, as I told you, reassociate after acting as a template for the new strand, thus restoring the parental double helical structure, right? And the newly formed strands join to form the helix. So this will be the daughter strand, and this will be the parental strand. So this type of replication is known as conservative replication. Now, talking about the last one, that is the dispersive type of replication. Here, the parental strand are dispersed by the name itself, right? It is going to get dispersed into two daughter DNA helix, into the two daughter DNA helix. Now, so that each strand is formed of transform of some part of parental and some parts of new DNA strands. So basically, if you see, you have a double helix. Okay, now here it will be dispersed, right? So let me just draw. This is the parental strand, then daughter strand, parental strand, then you have the daughter strand. Okay. It should be like this. Parental strand, then you have the daughter strand. Parental strand, you have the daughter strand. It will be actually dispersed in here. Okay, it will be like this. Okay, So this is how the dispersing method of DNA replication looks like. So we'll be focusing mainly on this semi-conservative methods. So this is all about an overview about the DNA replication. Thank you for watching.